afternoon, honorable judges and fellow participants. My name is Ji Wong.
And I'm Celine.
And we will be presenting to you today on the topic of viruses in therapeutic treatments and biotechnologies.
So we'll be starting off with the introduction, which is section 1. We will be defining what a virus is, the properties, the risk, and importance of a virus. Then we'll move on to our actual topic itself, which is virus application in therapeutic treatments and biotechnologies. Then we'll discuss the bioethics and consequences. Then we will conclude and show you our works cited.
So the first section is the introduction. So what is a virus? A virus is a microscopic infectious microbe with a segment of nucleic acid seen in the form of RNA and DNA. Whether a virus is considered a living organism or a non-living entity is still argued on about by modern scientists to this day, but many argue and agree that a virus is a non-living entity because of its lack of basic cellular functions and inability to reproduce on itself. It consists of a nucleic acid and a protective layer of protein, and it can only replicate itself by infecting and amalgamating itself into a host cell, in which it inserts its genetic material into the host cell, meaning that whenever the host cell reproduces, it makes copies of the virus. So there are several different kinds and types of viruses. However, most viruses take on the following physical properties. Viruses are microscopic, meaning that they fall under the size range of 30 to 50 nanometers. They have characteristics of non-living things, meaning that they lack cells, so they do not have a cell wall. And in place of a cell wall, they have a capsid or a nucleocapsid, which acts as the innermost layer of protective protein. The structure of a virus mainly consists of a nucleic acid surrounded by the innermost protective layer of protein known as the capsid, and the outermost layer of the virus is the envelope, which consists of envelope proteins, membrane proteins, and spike proteins. So viruses, as aforementioned, can only replicate itself by infecting a host cell, in which, whenever the host cell reproduces, it creates copies of the virus. Viruses have the ability to replicate itself at rapid speeds and rapid amounts, meaning that the cells can die out and possibly even lead to infection of other surrounding cells. Due to the very rapid speed and rapid amount that the viruses replicate at, human populations are more susceptible to virus-related outbreaks like epidemics and pandemics.
So now we'll be talking about virus application and therapeutic treatments and biotechnologies. So for centuries, viruses have been used in biotechnology, such as with vaccines. However, recently it's been used as material-carrying vectors, such as with viral vectors, agricultural applications, and using viruses to produce desirable characteristics, in nanotechnological applications, in metal deposition and nanomaterial production, and there have been other instances of viral application in other industries like pharmacology, cosmetics, electronics, and other industries.
Today, the main biotechnology we'll be focused on is genetic technologies, in which the three main primary applications of viruses are in gene therapy, oncolytic virus therapy, and vivo gene editing. So as a brief timeline of the major applications, in 1990, the first approved gene therapy was performed by W. French Anderson in 1990. In 1996, the first approved clinical trial of oncolytic virus therapy was performed, and in 2009, CRISPR, which is a revolutionary gene editing tool, had been released to the public. So first, what is gene therapy? Gene therapy is a form of genetic technology which involves the use of therapeutic genes to restore or recover from a genetic-related disease or illness. In this instance, viruses are used as vectors, which will carry the therapeutic gene into the cell, which will then be injected back into the patient in order to be used to produce the desired protein. Viruses in this instance are genetically modified so that no harm is spread to any cell, and that is unable to infect a patient's cell by using its own genetic material. The benefit of the virus in this specific instance is its ability to integrate itself. So advantages of gene therapy include the fact that it is a promising method of treating a variety of diseases, such as cystic fibrosis, heart disease, AIDS, etc. In few cases, this is very life-saving, as there are certain treatments in which surgery and drugs aren't very usable. And with more testing and research, this could become the treatment for the most devastating genetic disorders. However, alongside these advantages, disadvantages always follow along, with the big economic price of $2 million required to complete this procedure. There are also ethical disadvantages, which shows that people in low socioeconomic classes who may need this treatment as much as possible aren't able to afford it. And there are also safety concerns regarding this procedure, as there are multiple technical difficulties included, in which there are possibilities that the virus does not find the specific cell that it's supposed to amalgamate itself into. The next technology that we are focusing on is oncolytic virus therapy. Oncolytic virus therapy is a type of immunotherapy specifically targeted towards treating cancer. The viruses are engineered into oncolytic viruses, which intend on attacking and targeting and attacking tumor cells. Upon affecting a cell, the cancer cell will burst and it will release cancer antigens, and the benefit of this virus is to integrate. Advantages of this technology is that this is a treatment for cancer. The fact that this is a treatment for cancer is a big advantage, because cancer is one of the leading causes of death. Annually, there are 18 million people estimated to have been diagnosed with cancer, and of which, unfortunately, 10 million people pass away from. So the fact that there's a possible treatment of a leading cause of death worldwide is a revolutionary innovation in this field. However, disadvantages are that since this is a very new treatment and technology, there are possibilities that these oncolytic viruses could potentially miss-target and attack healthy cells that are not cancerous, which obviously poses health issues. There are also risks of carrying infection, and likewise to gene therapy, this is very expensive, and only one oncolytic virus to this day has been approved by the FDA.
So our third example is vivo gene editing. This is when gene editing is done inside of a living organism. This is as opposed to in vitro gene editing when it's done outside. So this encompasses various biotechnologies such as CRISPR-Cas9, which is a revolutionary genetic modification system, and it generally works by selecting a target gene, delivering the necessary editing components, modifying a gene, then repairing the gene. So there are many applications for this technology in human health, agriculture, animal models, and conservation. So the advantage of vivo gene editing is the fact that it has the ability to cure genetic diseases, which is a very pertinent problem in our world today. And it's also better in contrast for in vitro gene editing, because it's able to simulate the human system more in comparison, which allows it to be more transferable to humans. Furthermore, it has major, as mentioned, humanitarian, medical, and agricultural implications. However, in using in vivo gene editing, there's a lot of animal models used, which comes with a lot of ethical questions. Although we acknowledge that animal models should not be used in most cases, it's very imperative for them to be used for transferable treatments to humans. So furthermore, preparing these animal models are very expensive. They require a lot of time and resources. It's also, the technology is very expensive itself, and there's a risk of unintentional change, which may cause genetic mutations as well. So section 3 is bioethics and consequences, where we'll talk about any controversies and importance of bioethics. So bioethics is biological ethics, and it's very important because even as scientists, we have to consider the ethical implications of our actions, as the goal of scientific innovation is for the betterment of our society, and therefore it should not be detrimental or harmful to such. There are 4 factors or principles of bioethics, which is autonomy, control over oneself; beneficence, which is doing things with good intention; non-maleficence, which is an obligation to not harm anyone; and justice, which is fair judgment. So So viruses pose a lot of biosafety risks, especially with viral vectors, which is a primary form of viral application. So viral vectors are used in a lot of biotechnology, and although they are engineered to be replication deficient, which means that they can't necessarily divide and replicate, there are instances where they may reacquire the ability of pathogenesis, which allows them to spread further. And this allows them to potentially infect cells and therefore cause cell injury or cell death. There are two major disadvantages of viral applications that we talked about throughout this presentation. The first one is genetic mutation. There's always a chance that it may cause genetic mutation, as there's unintentional changes to occur to the genome, and this may cause off-site or on-site mutations, which are detrimental to the organism and the cell.
The other major disadvantage of viral applications in biotechnologies is the economic impediments. So there are economic disparities included in the application of viruses in biotechnologies because this current method of using viruses is prohibitively expensive. So this accentuates the current difference in social gap and wealth, and it essentially means that only the wealthy or upper class men are able to afford this technology, which is a major ethical drawback. And furthermore, high expenses and funds are required in order to achieve development and research in this area of research, which means that a lot of expenses are required. There are also controversies regarding each of the examples of technologies we had mentioned before. A controversy regarding gene therapy is safety. So as I had mentioned, gene therapy is quite new, thus there are still quite a lot of safety drawbacks, which include attaining allergic reactions, immune response, and the worst possible imagination, death. And one of the more famous cases is the 1999 case of Jesse Gelsinger, in which he was involved in a gene therapy trial in its early stages, and he unfortunately passed away from a genetic liver disorder due to the failure of gene therapy. Likewise to oncolytic virus therapy, this is also a very new treatment, meaning that there are still a lot of safety testing and procedures to occur. However, like Celine had mentioned earlier, it is impossible to gain full control over a virus, meaning that it has the ability, it's impossible to tell if it will entirely target a cancerous cell, and there are possibilities that it attacks a healthy cell.
So most controversies with vivo gene editing is in the unregulated gene modification of the human germline, which is basically when a gene is mutated, a gene is modified in a trait where it is heritable by future generations. So an instance of this, which is the most notable instance, was in November of 2018, where a Chinese scientist, Jiang Kuihe, modified twins to be HIV immune. Although he declared that his results were successful, this was an unprecedented event in scientific history, and it challenged many notions that we had about bioethics. This raised intense discussion and sparked a lot of arguments over the bioethics of genetic modification and the use of viral applications, which is why this is such a controversial event. So there's been a lot of biotechnological regulation due to these type of events. So many applications of viruses are still being tested and still in their experimental phases, which is why these are still being discussed. So in 2020, a study was done in 106 countries where genome editing policies in early stage embryos for IVF were examined, and out of these, 75 countries illegalized germline editing, and none of them allowed explicit permission for early stage embryo modification. In the UK, they passed a genetic technology, which is a Precision Breeding Act, which allowed the genetic modification of crops to address food insecurity. And in the EU, they plan to heavily regulate CRISPR-modified crops to the risk of genetic mutation. Furthermore, in the UK, the fertility agency HFEA deemed 3 parent babies to be morally permissible as long as it's in the interest of the future child, and as long as it doesn't add to inequality. Therefore, the first 3 parent baby was born in the UK. And on a larger scale, the WHO recently made some human genome editing regulations. This was made upon hundreds and thousands of discussions with hundreds of experts. And this is basically a plan to make a small expert committee, convene multi-sector stakeholders, and lead